This is the last and final video that you need to watch to understand how to use DNA Subway. This is video number 4, how to match your DNA sequence to a species. Before jumping into DNA Subway, let me point out the terms that you'll need to understand before matching your DNA sequence to a species. To know those terms, go on up to the DNA Analysis Word Glossary. Here, you can see the terms that it's necessary for you to understand to use DNA Subway to match it to a species. The first term you need to understand is a species match. The second term you need to understand is alignment length. And the last term to understand is what is called the mismatch score. Make sure to read about each of these terms before going into DNA Subway to match your particular DNA sequences to a species. Now I'll go ahead and open DNA Subway so we can match our DNA sequences to a species. After you've viewed your DNA sequence, you're going to click on the button that says BLAST TN. When you click on this button, it will open up to both of your DNA sequences. In this case, I'm looking at section A, team 2, sample number 1. I'm looking at this second sequence, section A, team 2, sample number 2. To match your DNA sequences to a species, you click on these little buttons that say BLAST. Once you click on the BLAST, it will open up a long list of possible species that could be matched to your particular sequence. In this row right here, you'll see what is called the alignment length, the vocabulary term that you need to understand. In this row over here is called the mismatch score, which is another vocabulary word I pointed out that you need to understand. Over here are the different species that could potentially be matched to your particular seafood sample's DNA sequence. Check out each of the species matches by clicking on the blue link. After clicking on the blue link, an Encyclopedia of Life window will open up. To find out more information, click on the link. After clicking on the link, it will bring you to the website Encyclopedia of Life. Here you can see that one of the species matched to this particular sequence is called the silver-striped round herring. You have the scientific name at the top, and you have the common name at the bottom. If you'd like to learn more about this species, you can scroll down and read what the Encyclopedia of Life website has to say about this particular species. In this case, it says usually an inshore schooling species inhabiting relatively clear waters of coastal lagoon and seaward reefs. Scroll down this page to read more information about this species. Going back to the DNA Subway website, you can visit a bunch of other species that are also matched to this particular DNA sequence. So for an example, this is a different species compared to the one that we just looked at. By clicking on the link, you can visit the Encyclopedia of Life page to learn more about this species. To figure out which out of this list of species is the best match to your DNA sequence, meaning what your seafood sample most likely is, to figure that information out, you want to look at the two scores of alignment length and your mismatch score. Once you compare these two scores together, it can tell you the species on this list that is best matched to your DNA sequence. After you find this information out, you want to make sure to write it down because this is the answer to your research question. Just to show you what it would look like if you tried to match a DNA sequence that has low quality to a species, this is what it would look like. Our sample A21, as you can see, has very low quality. If we tried to do a BLAST search that would try to match it to a species, this is what you'd see. You would have a screen at the bottom that would have a message, and you would actually have a blank list of any species that could be matched to your sequence. The reason why we are seeing a blank page is because sample A21 has extremely low quality. That means that it is not trustworthy for data, and we cannot match it to a species. Unlike our other sample, A22, which we could match to a species. You want to make sure to find the correct species match on this list, again, by using the alignment length and mismatch score.